Hi, everyone. We're going to begin a new unit today, which is going to be on plants. This is topic 5 out of 6 in the junior IB. So within plants, I want to just, do a little bit of a review on transpiration. And we know that transpiration is the evaporation of water, right, through the leaves of plants. So we're going to take a look at that today. So let's start by, kind of looking back at photosynthesis from pre-IB biology a couple of years ago. We know that green plants that have chlorophyll in them, will use the energy from the sun, that light energy, in order to turn carbon dioxide and water into oxygen and energy. So we're going to look at exactly how that occurs within the plant. And then we'll take a look at a couple of short videos that will not only, first one will explain the structure of a leaf, and then the second one will look at how we use a potometer, which if we were together in a lab, we would be able to build our own potometers and go from there. All right, so now in order to really understand photosynthesis and what is going on, we see these cells here, and we see these kind of guard cells that are harboring what we call a stomata. And a stomata is kind of like a pore in the leaf of a plant. This is the epidermis of the plant here, you can see. And inside some of these cells are what we would consider to be guard cells that allow water and things into and out of the stomata. So the stomata is able to open and close. So everything has to go through this stomata. We look at it a little more closely. Here we see the stomata or pore. Here it is open, and over here it is closed. We see these kind of long, oblong, I guess, what we call guard cells, and those kind of determine what can come in and out of the cell, right? We want carbon dioxide to be able to come in, and we want oxygen to be able to exit or diffuse out. So all those things have to happen in our cell. Now, if we're looking at the epidermis of the cell, we see we have to have this gas exchange that occurs. So, and all that has to go through the pore or the stomata of the plant. So carbon dioxide must come in, and of course the light energy is important here, and water diffuses out in transpiration, as well as the oxygen gas. That's where our gas exchange comes in, right? Carbon dioxide gas for water vapor and oxygen gas. So that gas exchange becomes extremely important. And when we're looking at that water loss, that's what we're talking about when we talk about transpiration, right? The evaporation of water through the leaves of plants. And with this, we're going to see, carbon dioxide usually comes in from the bottom or the underside of the plant, and then our oxygen leaves from the upper portion of the plant. So as water leaves the plant, that's where our transpiration comes in. But remember, our water has to move against the flow of gravity, right? So we have water coming in from the ground if there's rain, or if we water a plant, whatever it happens to be. That water gets pulled up through the roots, through the stem, from the underside of the plant, through the veins here of the plant, and it's moved onward and upward. Can't see that. All right. Most of that water, 97% of it that comes through the roots, ultimately gets lost up here in transpiration, right? So only about 3% of that water that's taken in is actually used in, running the metabolism of the plant itself. Kind of look here again, we have that pipeline that is going to move our water, and again, carrying other things in through the plant, out to the leaves, and eventually the transpiration of the water. Right? Carbon dioxide in, oxygen out. That's where the gas exchange. Now, there are two things that we look at that affect transpiration rate. One increases the rate of transpiration, that's temperature, and one decreases the rate of transpiration, which is humidity. So if we're looking at these in an experimental fashion, here we see our rate of transpiration, and on the x-axis, we have our temperature. So as it gets hotter, as our temperature increases, our rate of transpiration will increase until we get to a point where the water has it evaporates away, and we reach that plateau. The rate of transpiration increases until we hit that plateau. Now with humidity, it's just the opposite, right? Here's our rate of transpiration on the y-axis and our humidity levels on the x-axis. So as our humidity goes up, our rate of transpiration is going to come down, right? Because as we are giving off more and more water from the plant, that water has to diffuse into the atmosphere. And water, of course, diffuses down a concentration gradient. So if there becomes more water in the atmosphere than in our plant, transpiration is going to stop. And that's what we see happening here, right? As our humidity increases, our rate of transpiration starts to come down until that concentration that's outside, the water concentration outside the plant tends to be then greater than what is on the inside of the plant. Now, in some extreme places, we do have adaptations that plants have to make. So if we're looking here at desert plants where we have very little rainfall, we see that there is no leafy plant here because the desert plant has to be able to conserve water. So the larger the surface area, the more transpiration that's going to occur. And in a desert, of course, you don't want to have transpiration occurring. So we call these environments extreme environments. So the organisms that inhabit them are extremophiles. In this particular case, we're looking at what is called xeriscaping instead of landscaping, right? Putting green plants in. Sometimes people will put desert plants in or use rocks or things like that rather than grasses to be able to cut down on the amount of transpiration that's occurring. So we see the adaptations, right? Smaller surface of the leaf, so there's less transpiration. Really thick, waxy cuticles. Most of the cuticles on a plant are going to be very thin, but in desert plants, they're going to be much thicker, right? Again, to hold that water in. The stomata tends to open at nighttime when the temperatures are cooler. Again, remember, hotter temperatures, more transpiration. So cooler temperatures, less transpiration. And that's what we want to see. And then the stems being thick and able to store a lot of water. So that's kind of what you're going to see in your desert plants as far as getting them to hold water and cut down on transpiration. Now, the other extreme that we'll see sometimes are what we call halophytes, or plants that can tolerate high saline soil, so lots of salt. These are salt lovers. And consequently, their adaptations, are going to have, again, smaller stomata. That's what the salt is going to do. It's going to pull the water out. Longer roots, so that those roots, the tentacles of the roots, can go out in search of water. They'll have structures within their tissue that are going to be able to take salt out of the plant. And we see very small leaf size, so that the leaf is not transpiring as much. Now, the last piece of this is a look at our potometer, and we see the setup here. If we were in the lab, we would be using leaves or stems from the laurel plant. It's a very common plant, and it's very good for testing transpiration. So we do have potometers, and we would have set these up. The diagram shows what our apparatus here looks like. The key is to have a good seal over here where the plant is, so that transpiration is only occurring through the leaves of these plants, and it's not coming out of a pore connection here. We have water. We have our calorimeter here, our capillary tube, that's going to measure how much transpiration is occurring. And we can do that one of two ways. We can follow a bubble. We can start with a bubble and see where that bubble transfers to, or we can put food coloring or something like here in the water reservoir, and we would watch then the change in color, and that would give us a good indicator, rather than trying to follow the bubble. That a lot of times is one of the better things for us to do. So what we're going to do now is take a look at kind of a lab, how we would set this up. And there's basically two ways of doing it, and we'll show you both ways in the demonstration. And then there's a short activity on the structure of a leaf. That's it for today. Thanks, girls.